Good afternoon, everyone. It's my pleasure to welcome you all to the United Nations for the first ever 3MT competition. You all know what that means. The UN, we are very pleased to partner with Columbia to organize this event, an event with participants from 7 Ivy League institutions. I would not name all of them, but you know who they are. And this is a pleasure that we are able to do it. The objective of this from our side is that how do we bridge the gap between the academic ivory towers and the real world. You would go into the real world and would want to hear your ideas, and good ideas, as we believe, should be self-evident. If a good thesis, if you can explain it in 3 minutes, and if we understand it clearly, that means that we should be able to understand the implications. So, and you have the rules. I will not go into the rules, but just I think I want to just flag one point that why this event is important, because if you think about Albert Einstein, he didn't have 3MT. And if he had 3MT, probably he would have done much better in terms of selling his ideas to the rest of the world. Because he had to wait about 20 years after his PhD dissertation to really convince the real world that his ideas were actually quite valid and relevant. But better late than never. At least it worked for him. And I know that many of you have great ideas, and we have 5 distinguished judges, and we would have their considered views. Also, you have the opportunity to vote as audience. That would also give us an additional sort of perspective about the messages that you have in 3 minutes. And this is a very sort of exciting opportunity for us as we listen to you. We look forward to the clues and to how they actually can make a difference in the real world. And without further ado, I will request Andrea to welcome you as well from Columbia University. Thank you.
Good afternoon. I'm Andrea Solomon, Vice Dean of the Graduate School of Arts and Sciences at Columbia University. And welcome to the 2019 Ivy 3MT competition. Columbia is fortunate to be located in the city of New York, home of the United Nations, and we're privileged to be able to co-host this extraordinary competition. We welcome our friends across the Ivy League. Thank you so much for being here. I know many of you have come from very far away. This event is modeled on the University of Queensland's 3-minute thesis, an international competition in which more than 600 institutions participate. In today's iteration, local winners from Brown, Columbia, Cornell, Dartmouth, Princeton, University of Pennsylvania, and Yale will compete to present their PhD research in 3 minutes using only 1 slide. In this inaugural group event, we celebrate the scholarly achievements of PhD students from across the Ivy League, who in a few moments will be showcasing a range of skills, including critical thinking, data visualization, and written and oral communications. We're all enriched today by learning about the intellectual passions of these incredible students. Thank you for sharing your exciting work with us. It is now my great pleasure to introduce Eliot Harris, Assistant Secretary General for Economic Development and Chief Economist here at the United Nations. Mr. Harris is an expert on international economics and development policy analysis. He has extensive experience in the design of macroeconomic policies and its application as a central instrument for the reduction of poverty and for resilient and sustained economic development. Mr. Harris.
Thank you very much, Professor. Good afternoon, everyone. That sounded very impressive, didn't it? All that stuff that I did. But it's a little bit intimidating when I looked at the slides that are going to be presented in a moment. The future's in good hands, let me put it that way. A little game that I'm going to play. My son, who's 14, is going to be very interested in one of those slides. See if you can guess which one. I'm very happy that we're able at the UN to host you for this event today and to welcome this inflow of new ideas that come from today's PhD students and tomorrow's leaders. We at the Department of Economic and Social Affairs, our focus is on promoting socioeconomic development, sustainable development, and we try to devise policies that will assist the member states in making the right choices, using the resources that they have as effectively as they can so that we can reach the sustainable development goals that we all subscribe to. Those policies have to be based on rigorous evidence-based analysis, and that, I think, is the approach that we try to take as much as possible so that we can give the member states something concrete to work with. Now, in that effort, we're always looking for new, innovative ideas. Not just the ideas that we generate in-house, but also an influx of ideas from outside, because that influx is what helps to ensure that we are actually looking at the right things, that we are addressing issues that are of relevance to the people out there, not just to the member states here in our ivory tower. We have our own ivory tower, don't we? Yeah. And those ideas, they come, of course, from academia, from civil society, from scientific institutions, and from other multilateral organizations. Now, because of the nature of the Sustainable Development Agenda, I'm hoping that, I won't go into it, but I'm hoping that you all are at least passingly familiar with that. We are talking about making progress in a whole range of interconnected areas, and consequently we need ideas that come from a broad range of disciplines. It's not enough for us to sit here and talk about economic policy or social policy. We need to be talking about the science. Hamid here runs what we call the, how do you call it, the quarterly?
Technology quarterly.
Yeah, quarterly technology publication, and that's one of the things that we know is going to affect the way we live our lives already, and much more so in the future. So that has to be a part of the kinds of policies that we think about. So we need to look not only at economics or at the social sciences, but we need to look at the humanities, we need to look at mathematics, physics, arts, engineering, and so on. So I'll be listening very keenly. I'll have to report back to my son about which one of the slides won. But I'd really be keen to see what your research has revealed. And before we start and before the judges get to do their thing, let me congratulate all of today's participants and wish you all the best of luck. Congratulations. Thank you.
Thank you, Assistant Secretary General. I'm Richard Slusarczyk, Associate Dean of Academic and Student Affairs at the Graduate School of Arts and Sciences, Columbia University, and your MC for today's event. Before we start with our finalists, I would like to acknowledge our judges. Salim Hasham, Partner McKinsey & Company, Maria Angela Paralancourt, Senior Economics Affairs Officer, United Nations Department of Economic and Social Affairs, Laurel Patterson, Team Leader, SDG Integration, United Nations Development Program, Alex Sarian, Acting Executive Director, Lincoln Center Education, Lincoln Center for the Performing Arts, Mariette Westerman, Executive Vice President for Programs and Research, the Andrew W. Mellon Foundation. Judges, please stand. Thank you. Today's competition will begin shortly with presentations from 14 finalists. Each finalist, as we noted, will present for 3 minutes using only one slide. The top 2 presenters, according to our judges, will be awarded prizes of $1,000 and $500. Audience members, keep in mind your vote counts as well. Following the presentations, we will provide instructions for you to cast your vote. Make sure to cast your vote for only one finalist. During today's event, you are welcome to take photographs, but please do not use a flash. We encourage you to join us on social media by posting your photos with the hashtag IV3MT2019 as printed on the front of your program. And now, we will begin with our first presenter. Leon Wong, Cancer Diagnostics Using Light and Sound.
Hello, hello. Well, thank you so much for the most welcome introduction. The first thing that I'm going to ask our audience to do for me is to indulge me on a thought experiment. Imagine you're back in high school and you're getting ready to ask your crush to prom. You may have anticipated this moment for maybe an entire year now, and you finally muster up the courage to ask that million-dollar question. And what is the response that you get? Let me get back to you in 3 weeks. But imagine a different scenario where a patient is asking a doctor, hey doctor, do I have cancer? Did you know that the average time it takes from the first doctor visit to the start of cancer treatment for an average cancer patient is a staggering 62 days? 2 months. What are those 2 months like for a cancer patient? Can you go back to work? Can you spend quality time with your family? Or are you wondering day and night, unable to sleep, if your cancer is getting worse? Is it growing? Is it spreading? Is it getting harder to cure? Answer is, it's actually doing all three. So why does it take so long? Part of the reason is because all cancers are different. And for a doctor to determine the best type of treatment, they would first have to determine what type of cancer a patient has. To do that right now, essentially there is an operation called a surgical biopsy, where you get a piece of the tumor that's taken out of you and sent to a research lab somewhere in Boston or California for tests to be done. That process can take up to 3 weeks. So we're approaching this problem from a different angle. The question that we're asking is whether we can combine already pre-existing medical imaging technologies with our lab's technologies on nanoparticle systems to give patients an immediate response, to give patients immediate results to their diagnostics. So, as you can see in the slide above, we're able to leverage our lab's nanoparticles in designing many types of nanoparticles that each target to a specific type of cancer. And by tuning the color of these nanoparticles, they can only react to a different color of light. And leveraging this photoacoustic imaging technology, we can then generate these very colorful images, essentially telling a patient and a doctor not only if they have cancer, but also what type of cancer they have, all in one go. We have so far tested this technology in a mice tumor model for lung cancer, but we hope to be able to expand this technology in other areas of cancer as well. Thank you so much for your time.
Her topic is Oceans on Early Mars.
Have you ever looked up at the night sky and wondered, is there life out there? Or are we alone? I've been interested in these questions since I was a child. For my PhD, I'm asking, was Mars ever habitable with liquid water and a warm climate like the Earth? And could life have ever formed there? One way that I learn about what Mars used to be like is by analyzing ancient geologic features that are on parts of the surface that have been preserved for a very long time. Then, knowing what these features are, I use mathematical models to constrain the climate conditions in which they formed. One famous feature class on Mars are the dried rivers, which imply that there was liquid water on the surface of Mars 3.5 billion years ago. And this is exciting because liquid water is one of the key requirements for life, but it's not the whole story. Since we want to know if Mars was ever habitable, we also have to know about climate, not just the presence of water. And unfortunately, rivers can't tell us much about climate because they can form in very cold and very warm places. But rivers aren't the only features preserved from this time. Potential shorelines and tsunami deposits suggest that there may have once been an ocean that encompassed the entire northern hemisphere of Mars. And my research shows that this ocean could be the key to unlocking the climate. You might be thinking, what can an ocean tell us about the ancient climate on Mars? Actually, a lot, because this ocean can only form under very strict climate conditions. Let me explain. This ocean was likely sourced from groundwater. In order for groundwater to be released from the subsurface to the surface, the local temperature has to be above freezing. If it's below freezing, the frozen ground will trap that water beneath the surface. So we know the local conditions of the climate required for this formation of the ocean and the location at which it formed. In my research, I use multiple mathematical models to paint the picture of what the climate was like on the entire planet when this ocean was in the northern hemisphere. In other words, I'm asking, if you were on Mars billions of years ago, would you have been relaxing on the beach in a bathing suit, like in tropical regions on Earth? Or would you have been observing half-frozen waves splashing into icy shores, like in Antarctica? My research finds that the local conditions required for the formation of this ocean could have persisted in the topographically lowest parts of the northern hemisphere, even if the average global temperature was 20 degrees below freezing. And that's the average temperature in Antarctica. So maybe it wasn't beach balls and bathing suits, and we should start envisioning Mars as a place with very cold oceans. And if life ever did form there, maybe it would have had to have been organisms capable of surviving harsh conditions. This link between climate and geology is teaching us a lot about what Mars used to be like and helping us answer those questions we ask when looking up at the night sky. Thank you.
Sinrang Chua, Guarding Against the Climate Impacts of Air Pollution.
The acrid smoke clung to our hair, our skin, our clothing. It was forest fire season, and the wind had blown the smoke from the neighboring rainforest over the city of Singapore, where I grew up. My dad knew just what to do. He reached into the drawer, took out the surgical mask, and then we were safe. Or were we? The fact that we can see air pollution implies that it interacts with sunlight. Sunlight can bounce off these particles, and these particles can also absorb sunlight, using this energy to heat the atmosphere. This means that the impact of air pollution doesn't just harm our health, but it has large climate consequences. This heating is so strong that globally, it is 70% of that caused by carbon dioxide, which, as we know, is the strongest anthropogenic greenhouse gas. Carbon dioxide concentrations are easy to model. They are evenly spread in the atmosphere. In contrast, smoke particles are highly variable, being strongly concentrated in areas of industrial activity and forest fire burning. In my thesis, I'm trying to understand how these varied smoke patterns affect climate extremes. To do so, I make use of high-resolution computer models of the Earth's atmosphere and climate. When we heated the atmosphere uniformly, we find that it makes it harder for raindrops to condense and form. This allows moisture to build up in the atmosphere. When a storm does get to form, it finds it has much more moisture available to it. There's more to pick up, more to throw out, leading to greater risk of floods. Does it matter where we place this smoke? We switched the horizontal distributions of smoke and found that the atmosphere, as a fluid, is so efficient at mixing heat that even forest fires burning far away in Southeast Asia can affect us right here in New York City. What we did find, however, is that the vertical distribution of smoke particles is highly critical for its climate impacts. We are used to worrying about the air pollution at the surface, which we can see and feel and touch. But because raindrops form high up in the atmosphere, it is the smoke lofted up above that actually has the greatest climate impact. The mask can only protect from the threats right in front of us. It cannot protect from the distant threats from far away and high above. My research is helping us to uncover these distant threats so that we can design better policies to safeguard our children and their future. Thank you.
Alexander Kasnel, Engineering Collagen for Better Materials.
So collagen is nature's favorite building material. It's the most abundant structural protein we have in our bodies. It's the most abundant one in the entire animal kingdom, actually. You have it in your skin, your bones, your eyes. It's in countless tissues in your body. They all come from that same molecule you see illustrated there. And because of its versatility and robustness, how abundant we have it in our bodies, we've been able to use it for centuries as a biomaterial. You can use it to make biodegradable sutures for surgery. You can make biocompatible wound dressings. You even go into the drugstore, you see collagen skin creams on the shelf. They're all coming from that same material. But the problem with the status quo of collagen materials is twofold. First, natural collagens come from animal sources, meaning we have to use extensive purification and processing to make them safe for humans. And the problem is, even when you do that, the implantation process can sometimes introduce an immune response in the patient. They're not totally safe. The other problem is that synthetic collagens that attempt to circumvent that issue, they're very expensive to produce and they're not very stable. They're rapidly degraded by the enzymes in our body. And so by being bioengineers, we look at this problem and say, how can we make collagen into a better material? And to do this, we had to use chemistry. Now stay with me, we can do this, I promise you. So it's made of 3 building blocks, collagen is. It's proline, hydroxyproline, and glycine. All we did was we went into the lab, we synthesized a non-natural mimic of glycine called azoglycine. We inserted it into a synthetic collagen. What that does chemically is creates additional bonds in the collagen molecule, makes it more stable, and makes it resistant to degradation by those enzymes we have in our body. So effectively what we did is use standard synthetic chemistry techniques and low-cost materials to make a synthetic biocompatible and low-cost material, a hyperstable version of collagen. And my PhD is to take that material and use it to make, for example, wound dressings that won't break down as quickly. You can use them for the treatment of chronic wounds and for the sustained release of a drug payload. You can use them to make biological imaging agents for studying natural tissues and for diagnostics. You can even use them for another big scary word called tissue engineering, which is basically making model systems of the tissues we have in our bodies to create diagnostics, for example, to test a new medical treatment before we implant it into patients in an accurate way. And so that essentially is what bioengineering is. We're using scientific knowledge and engineering principles to solve the problems of our world, and that is my PhD.
Bailey Brown, Kinder Panic, School Selection and Parental Uncertainty.
When was the last time you were uncertain about something? As a teen, maybe it was waiting to hear about your college application. Now, maybe it's waiting to hear about a job offer or deciding whether to move to a new city. We all face countless decisions in life and can probably admit to feeling uncertain for some of these big decisions. Now I'd like to set a new scene for you. Imagine that you're a parent and your child is 3. At the daycare, you overhear parents talking about pre-kindergarten, and you begin to think to yourself, am I already behind? How will I make this decision, you might wonder? In my research on parents, I use the phrase kindergarten panic, or kinder panic for short, to describe this moment of uncertainty. I argue that this feeling is linked to the increase in public school options over the last several decades. Today, 2 out of every 5 parents has a public school option available to them. These options include district transfers, charter schools, and magnet schools. So these options provide more school choices for parents, but also place more responsibility on parents to make school decisions. To investigate this new responsibility, I interviewed 100 low-income and middle-income parents from diverse backgrounds in New York City. Each interview focused on parents' experiences applying to elementary school. I then systematically coded these interviews and immediately identified repeated trends of stress and anxiety. I found that parents struggled in 3 main areas: managing applications, understanding school admissions policies, and interpreting complex school data. So why should we care about the decisions parents make for their children? Well, for one, we all face decisions, and we have all felt how difficult some decisions can be. More broadly, the decisions parents make have long-term social consequences. Public schools of choice are expanding across New York City and the United States, and the decisions parents make in elementary school can shape how students are tracked in middle school, high school, and college. Beyond education, these findings shed light on decision-making as an important area of inequality. We tend to think of wealth and income as the most intuitive measures of inequality. But through the concept of kinder panic, I demonstrate how inequality is also experienced in the everyday decisions people have to make. When low-income parents have to invest more time and energy in everyday decisions like choosing a public school, this can perpetuate disparities between the rich and the poor. So the next time you have to make a decision, think about kinder panic and think about the way decision-making for elementary school and surely a host of other circumstances may result in unintended consequences. Thank you.
Mirav Salehi, Individualized and Task-Specific Functional Brain Mapping.
Imagine you're in New Haven. You want to walk to Yale's campus, but your phone's map is showing New York City. Most likely you'll get lost, or will surprisingly find yourself at Columbia's campus. Such mishaps happen when the city and the map do not match. A similar mismatch exists in the field of human brain mapping. The current methods in the field average data from everyone's brain and assign functional role to different regions to construct a single brain map to represent all. However, this brain map may not match with each single individual because all individuals are unique, just as cities are. Let's go one step further. Even within a city, our routes might change based on our task, like going to work or picking up groceries. The same holds for our brains. The routing of information in the brain changes based on the task we engage in. For my PhD thesis, for the first time, I challenged my field's long-standing assumption that there is a single brain map that works for everyone across time. I looked at brain images acquired by functional magnetic resonance imaging, or fMRI, and tried to understand what brain regions talk to each other and how this conversation changes as we engage in different tasks, such as playing a game or watching a movie. I developed an artificial intelligence technology to automatically translate these brain images into brain maps, like the ones you can see here. I applied this technology to brain images of 718 individuals and constructed their brain maps as they performed 8 different tasks. To my surprise, I observed these brain maps are unique to each individual, such that I could identify people with 99% accuracy just by looking at their brain maps. The brain maps are like fingerprints. I also discovered that these brain maps are not fixed, but they reliably reconfigure by task, such that I could predict with up to 97% accuracy what people are doing just by looking at their brain maps, which means I can tell what you're thinking about my talk without asking you, just by scanning your brain. My research has important clinical and social implications. Our personalized brain mapping technology could help doctors better understand individual differences in brain function and disease. This, in turn, could bring us one step closer to personalized medicine. And hopefully soon enough, you'll benefit from personalized diagnostics and treatment that's specifically designed for you, your body, and your brain. Just like the New York City's map that's specifically designed for New York City, but not New Haven. Thank you.
Teddy Samuelson, Yesuda-san, what makes a red potato red?
Growing up in southern India, all I saw were white potatoes. And so when I came in as an international student and went grocery shopping for the first time, I was shocked to see potatoes that came in all different colors. Providentially, I got into a lab that was studying pigmentation or color in potatoes, and so I was very excited. You know, we human beings have always been intrigued with color, and scientists are recently reporting that anything naturally colorful in your diet is beneficial for our health. You know, at this point, some of you are probably wondering, why does he have flowers on his slides? Well, these pretty flowers are actually potato flowers, and I'll get to it in a minute. So 10 years ago, the lab that I'm currently working in discovered the gene that is responsible to make red pigments in potatoes. And when we looked at this gene, we saw that it had multiple alleles. Now, most of you here know what a gene is. Let me tell you what an allele is. An allele really is a different version or a different form of the same gene. For example, take, for instance, eye color. All of us in this room have the same gene for eye color. But the reason that we have different eye colors is due to the different allele or a different version of the same gene that is expressed in each one of us. Now, going back to my potato alleles, we saw that one allele was able to make red pigments and another allele was not able to, which is labeled as the red allele and the non-red allele. And so when we looked at it in depth at the DNA level, we saw that there were certain DNA letter changes between these two alleles at certain positions. And so using genetic experiments, we swapped the DNA letters between these two alleles at the positions that were different and introduced them into a potato variety called Prince Harry. Now, Prince Harry on the left has pale blue flowers. And so when we introduced our alleles, we saw that the color of the flower changed from pale blue to purple. Why? Because pale blue with a little mixture of red should give purple. And that is exactly what we observed. And so through our experiments, we have been finally able to narrow it down to 2 DNA letters that are influential or crucial in converting a white potato into red and vice versa. In other words, we've identified 2 DNA letters that make a red potato red. Now, I'd like to suggest 3 things that we can do with this data. One is that this information will help us breed better quality red pigmented potato varieties, which are nutritious for us. Second, did you know potatoes are one of the most cheapest crops to grow? Hence, potatoes can be used as a wonderful resource for extracting natural dyes. And 3, this might seem a little far-fetched. Everybody loves potato chips, right? What if we could, in collaboration with food scientists, engineer potato chips that are actually pigmented and beneficial for our health? This is why I study red potatoes. Thank you.
And Norman Schiff, Writing Beyond Reading in Ancient Judaism.
Jews, ancient and modern, are often referred to as the people of the book being the Torah or Hebrew Bible. So scholars of writing in ancient Judaism have generally focused on the Bible, this central and elite and important book. I'm interested in the question of how writing affected the ordinary lives of ancient Jewish people. So I looked at a different kind of writing, specifically legal documents and religious amulets. I analyzed both ancient laws about how they should be written and actual archaeological examples from the Dead Sea Scrolls in order to find more about how they were used in ordinary Jewish people's lives. Let's take a look at what we have here. We have archaeological examples from the Dead Sea Scrolls of both ancient legal documents and ancient biblical verses. On the upper left, we have a marriage document from an ancient Jewish woman named Babata who lived in the years 100 CE. Because the caves near the Dead Sea were so very dry, it's preserved for us both the documents and the organic casings that were kept around them. So we can see that this woman kept her legal documents inside of a leather pouch. We should imagine that the documents spent most of their time in the pouch, and that Babata herself was probably unlikely to read them. Similarly, on the bottom, we have examples of an amulet. We have biblical verses on parchment, which would have been rolled into very small scrolls and put inside a leather amulet case, like we see on the lower right. We should imagine that leather amulet case closing from top to bottom, like a clamshell, and that it would not be opened. So in both cases, we have examples of writing that does something, but it does something without being read. And the archaeology itself shows us this. In my work, I also look at the ancient religious laws about how things should be written, which specify the kinds of parchment and inks, the shape of the letters that should be used. And I found something interesting. I found that the same laws that apply to the book, the Bible, that specify the way its letters should be written, also apply specifically to ancient religious amulets. Now, we might imagine that we would care about these letters because they're part of a book, the book, a Bible that we read. And yet, they seem to apply equally to amulets that no one reads. And I think this tells us something interesting, that ultimately we should look at the Bible, the book itself, as like an amulet, something that functions in ancient Jewish lives beyond the function of being read. Thank you.
Jess Savitson, using mini brains to model brain injury.
We're all familiar with the dangers of concussion, but what's going on with the actual cells in your brain when it gets injured? More importantly, how can we protect your brain from trauma, and how can we help it heal? To address these questions, my lab creates mini brains from brain cells. The mini brains have all of the neurons and the other cells that you would expect to find in a brain, but each one of these is actually about the size of a grain of sand. Now, it's important to make these mini brains in 3D because it allows the cells to more closely mimic the natural environment of our brains. They take on the shape, the density, the tissue stiffness that you would find inside of the body. However, because they are outside of the body, we have control over things like how many cells are in each mini brain, over what conditions they're exposed to throughout their development, over how hard they are injured. So while my colleagues are asking if the cells are alive and if they look like healthy cells, I take it a step further, and I ask if they're communicating like healthy cells. I do this using a technique called calcium imaging. Basically, I add in a special chemical so that whenever a neuron is active enough to bring in that extra calcium, it lights up. I take videos of the mini brains and use a series of computer programs to tell me which neurons, which brain cells are lighting up and when. All right, under normal conditions, the neurons form networks in pretty much the same way. They make those connections, they show synchronized activity, but then as they develop, that activity becomes more complex. And that's especially interesting because it's actually pretty close to the way that activity develops in healthy brains. What we're starting to look at now is what happens when we subject these mini brains to traumatic injury. To do this, I take a mini brain, place it in a device that's essentially a super precise scientific hammer, and impact them. Then I ask what happens to the cells afterward. In the future, we'll be able to test out a lot of different therapies very rapidly, which is especially important because right now there are no therapies for TBI. And that's the goal of my work, is for these mini brains to have real beneficial consequences for patients in the clinic. And I only had 3 minutes here, so I focused on traumatic brain injury, but we're also working on models of stroke and chemical toxicity. And the most amazing part, we get all this from a ball of cells the size of a grain of sand. Thank you.
Xiaopei Zhou, decoding the 2% that makes us human.
The DNA sequence of human and chimpanzee are 98% identical. But we look and behave so differently. What are the 2% difference in DNA sequences that make us human? Many of them are transcription regulatory elements. They are DNA sequences that do not encode gene, but determine when, where, and how much of each gene should be turned on or off. It's like playing a symphony. Yes, we need all the music instruments, the genes, but without the music sheet that denotes when, where, and how each instrument should be played, there's no symphony. Our understanding of transcription regulatory elements has focused on enhancer, the DNA sequence that drives the expression of the target genes. It works like a gas pedal. Could you imagine a well-functioning car with only gas pedal, but no brake? To solve that, the field has hypothesized that there's another kind of regulatory element, silencer, that will turn down the expression of the target genes. To study those hypothetical silencers, I developed a method to identify them based on their function. What's the function of silencers? Silencers work like a brake. Therefore, I would like to identify regions in the genome where higher activity reduces the expression of the target genes. To do that, I used the 2 copies of genome from our cell, one inherited from our mother, the other from our father. Based on the natural genetic variations between them, I can identify regions showing different activity that are representative of silencers. Think these 2 copies of genome as 2 highways that are very similar. The vehicles are the enzymes that transcribe RNA from DNA. The more traffic means the higher activity of transcription. Based on the traffic pattern, I can identify potential roadblock and their effect on the traffic. For example, stop signs slow traffic down. There's more traffic behind a stop sign and less traffic in front of stop signs. Using this high-low traffic pattern, I can identify the location of stop sign along the highway. Similarly, I can locate silencer and their target gene along the genome based on the activity pattern. Using this method, I've identified potential silencers. Next, we can start to ask, how do they look like? How do they work? And finally, we can answer the question, how do transcription regulatory elements create a difference between us and the chimpanzee? Thank you.
Hannah Shanehart, linking genes to brain in sensation and decision-making.
The glare of sunlight on a skyscraper, an itchy sweater, your coworker slurping down her tea. For people with ADHD, autism, and schizophrenia, even everyday sensations can be overwhelming or even physically painful, as described in this cartoon by an artist with autism. All 3 of these conditions affect the way the brain filters sensory information. And moreover, each of these also causes characteristic changes in decision-making. When I first learned this, I was really intrigued because ADHD, autism, and schizophrenia also all tend to run in families. In fact, scientists have started to identify specific genetic changes that increase the risk for these conditions. But what's missing is a description of how those particular genetic changes in turn affect the way the brain takes in and processes sensory information for decision-making. My project aims to fill this gap by describing a way a gene can affect the activity of brain cells as they're participating in a decision. And I do this using a cool model organism. It's kind of obscure, you've probably never heard of it, the young zebrafish. So why young zebrafish, I hear you all wondering? Well, they can be produced thousands at a time, which means we can do big, powerful experiments. They also share many of their genes and a basic brain layout in common with people, which means we have a good shot that our discoveries will be relevant to human biology. Also, at that young age, their brains and bodies are completely clear, which gives us a scientific superpower. we can watch the development and activity of their individual brain cells live. And finally, they have standardized and consistent behaviors. Now, I study the selection between 3 different behaviors in response to a loud noise. In other words, I'm looking at a sensory-based decision. Now, my lab screened through thousands of genetic mutations to find ones that affected a fish's bias on this particular decision-making task. And I was part of a team that tracked down the gene affected by one of those mutations. Now, my project focuses on watching the brain activity of fish mutant in that gene live as they're hearing that loud noise and making their decision based upon it. In other words, I am working to describe how brains and all of their diversity handle the world in all of its complexity. Thank you.
Christina Gilligan, Measuring Distances in Astronomy.
So knowing how far away something is, that's a pretty basic idea, right? It's a very important thing to know. How far away is the sun? How far away is the moon? But in astronomy, this is actually one of the most difficult and most controversial questions we have today. Astronomers and physicists are always fighting about how big is the universe? How far away is this other galaxy? And I'm hoping through my thesis to figure out a better answer to this question. But let's first start with a little thought experiment, because, you know, as scientists love thought experiments. So imagine we have our friend. Our friend has this very nice whistle. This whistle has the property of it always has the same loudness. No matter how he blows it, it's always the same hertz and pitch. So what you do is you have your friend stand, I don't know, about 5 feet away, and you figure out how loud is it now. Make him go 10 feet away. How loud does it seem now? We can make this relationship between how loud it seems versus how loud it is and compare it and figure out his distance. Now, this works if we know how far away he is. Let's say we send him somewhere really, really far away, like Dartmouth, for example. We don't know how far away Dartmouth is. So we measure the pitch or the decibels, how loud is it, and we can figure out the distance. Now, one way we do this is with this very, very special type of star that it's a variable star, so it gets bigger, smaller, and through getting bigger and smaller, it gets brighter, fainter, with a very, very specific frequency, a very specific period. Now, the cool thing about this period is it's actually related to how bright the star actually is. So measuring period is very easily. You just have to watch it go up and down, and I've spent a few weeks alone at an observatory doing this for dozens and dozens of stars. And we can calibrate these with distances of ones we already know. There's a few hundred of these that we've already measured the distance to. We compare its period, and now we can figure out this relationship. So in the little diagram at the bottom, we can use a telescope to measure one in a galaxy that's not even ours. This is a beautiful picture of the Large Magellanic Cloud, which is very close to our own galaxy. We can measure an RR Lyrae there and figure out its distance, which we wouldn't be able to do any other way. So through my thesis, I hope to figure out the distance to the LMC, which has very, very big implications. Excuse me. And hopefully, we can figure out how old the universe is, how big the universe is, and prove the astronomers right over the physicists. Thank you.
Sean O'Neill, The Art of Signs, Symbolic Notation and Visual Thinking in Early Modern Europe.
So if you or someone you love did not particularly care for algebra in school, then it might be comforting to learn that the famous philosopher Thomas Hobbes wasn't much of a fan either. Once, upon seeing a colleague's algebra book, he dismissed it as, quote, a scab of symbols. And yet such symbols appealed to many of Hobbes' contemporaries. In the years between 1500 and 1800, not just mathematicians, but from left to right here, dancers, chemists, astronomers, musicians, and stenographers all began to experiment widely with new symbols. And in each field, there were critics who denounced the practice as dangerous. My dissertation, The Art of Signs, Symbolic Notation and Visual Thinking in Early Modern Europe, asks the question, why did symbols both fascinate and frustrate early modern audiences? To answer this question, I traveled to archives and libraries in both Europe and the United States, where I studied old books, manuscripts, letters, lawsuits, and student notebooks, in which people debated the pros and cons of writing things down in symbols. What I found was this. The reason symbols so unsettled critics was that they evoked the very mysticism which many people at the time wanted to be casting off. During the scientific revolution, symbols looked to many like a step backwards, a form of intellectual cryptography that kept knowledge secret, as opposed to making knowledge accessible. However, advocates valued symbols because they enabled powerful techniques of what we would now call visual thinking. Just by looking at the size, shape, color, orientation, or location of symbols on a piece of paper, someone could quite literally see information more quickly and more easily than if that same information were described with words. And the more people recognized the value of such visual thinking, the less they worried about the seeming strangeness of the symbols themselves. Today, visual thinking is crucial to how modern science, government, and business operate. Every day we use charts, graphs, diagrams to render data intelligible and therefore also usable, such as this one on the right, which visualizes the changing levels of RNA in the cells of a fruit fly over the course of 2.5 days. It's been estimated that by 2022, the data visualization market will be worth $7 billion. We take visual thinking for granted, but as my dissertation underscores, this was not always the case. We had to learn to trust it. In an age before we had charts and graphs, people used symbols to visualize information. What's more, as they grew more accustomed to the advantages of visual thinking with symbols, they grew more accustomed to the advantages of visual thinking in general. So much so that when the chart and graph did first appear towards the end of the 18th century, the intrinsic value of visual thinking had already been well established. For centuries, people have used visualizations, be they symbols or something even more abstract, to both represent and to discover new knowledge. Who knows what we'll see next? Thank you.
Our final presenter is Rachel Brogue, Self-Driving Electric Cars.
Who would have guessed that Netflix would eliminate movie rentals, landlines would become obsolete, and we would not only be driving electric cars, but that they would be driving themselves? Like innovations from Apple and Tesla, technological advancements are changing cancer treatment as we know it. Currently, it's estimated there will be just under 2 million new cancer cases diagnosed in the United States this year alone. That's 5,000 cases a day and 9 new cases within the duration of my speech. Since tumors evolved from what once was a healthy cell, our incredibly efficient immune system fails to recognize them as a tumor. So how do we treat such a great number of tumors? We use harmful treatments like radiation and chemotherapy, which kills off healthy, vital cells. We essentially poison patients, a chance both doctors and patients are willing to take for a chance to save their lives. Instead of using toxic treatments, I study ways in which we can retrain the immune system to recognize the tumor. To do this, we take a sample of the patient's blood, similar to a blood donation, and infuse it with specialized receptors that specifically recognize the tumor. These reprogrammed cells are called CAR T cells, or CARs. CARs are infused back into the patient's bloodstream and vigorously attack the tumor in the way the body would naturally clear an infection. CAR T cell therapy is unlike any treatment we've ever discovered. I'm working to develop CAR T cells to be self-driving. Similar to a Tesla, CAR T cells need to be controlled. They need to be equipped with sensors, navigation packages, and the ability to respond to their environment quickly to ensure optimal safety and function. By using electrical modeling, I am working to accurately predict how CAR T cells respond within a patient and engineering the necessary sensors as biological switches so that they can respond to their surroundings and more efficiently clear the tumor. CAR T cell therapy clears tumors in a more natural way, not by poisoning patients, but by educating their own immune response. I hope to ultimately design a self-driving electric car. A car that doesn't get you to work, but ensures you're healthy enough to go to work. But we won't stop there. As we learn how to finely tune the immune system, we hope to be able to treat diseases like Alzheimer's, HIV, and more. Buckle up as we speed ahead full throttle, because we're engineering the unimaginable. Thank you.
Let's give all of our finalists a round of applause. Now, it's time for the audience choice voting. At this time, we would like you to cast your audience vote. Ushers, please raise your hands. If at any time you have a question about how to cast your vote, one of our ushers will be happy to assist. Remember, you can only submit one vote. Using your mobile device or laptop, please open up a web browser and type bit.ly/IV3MT2019. Select one finalist that you would like to vote for and hit Submit at the bottom of the page. We will keep the voting window open now for 3 minutes. Not the contestants. Okay, if I could have your attention, it's now time to announce the winners of the inaugural IV3MT competition. Thank you again to our judges. To start, the winner of the audience pick is Bailey Brown from Columbia University. Who presented on kinder panic, school selection, and parental uncertainty. Coming in second place, presenting linking genes to brain in sensation and decision making, Hannah Schoenhart from the University of Pennsylvania. And finally, I'm pleased to congratulate our first place winner, Marave Salihan. Thank you to all of our participants. And now, Andrea Solomon will say a moment to Eliot.
Well, thank you to all of the finalists for those really exceptional presentations. And I hope that somebody here was taking due note, because you managed to convey all of that information in 3 minutes, which is a talent singularly lacking among the diplomats of the United Nations. I found one of the presentations, I won't name it here, totally and surprisingly relevant to the work that we're doing here in the United Nations in the Department of Economic and Social Affairs. And it opened up an entire and utterly new perspective on an issue that we have found to be tremendously important, that we are actually researching right now. And I can promise that this has changed the way we're going to be looking at that particular issue going forward. I don't want to go into any detail about that, because I don't want it to say that, well, which one we like or which one we're going to use. But we're in the Department of Economic and Social Affairs, so guess. But that's the point, is that, you never really know when that little impulse to think about something differently is going to come, or where it's going to come from. And I think that's the real value of what we experience here today. It's particularly empowering to see young people like yourselves dealing with the problems of tomorrow with solutions that we can apply today. So thank you very much for my continuing education And let me hand it over to Hamid.
Thank you, Eliot. It was quite exciting, and I'm glad that I was not a judge, because as a judge, I would have faced a serious difficulty in choosing the winner and the runner-up. And also, I think it is quite fascinating about the spectrum of ideas that you brought forward. And many of them, as Eliot mentioned, some of them are directly linked to what we're doing, but also I can see the potential spin-offs of many of the ideas. Many of them have real application for sustainable development goals that we're pursuing in health, in education. All of them are very relevant. But also, I find it fascinating that, that the interest in brain, 4 out of 14 presentations were directly related to brain and neuroscience. I don't know whether it is because the brainy people are interested in brain or is because of something else. But it is quite interesting that we are, as we are looking at the outer world, the world around us as far as the Mars, or the distant planets and galaxies, but also we're looking at internally into our brain. And that's quite fascinating to see how we are in our vision for future, and how our challenges that we are trying to resolve are both within and outside. And that's also quite an interesting takeaway for me in terms of what we should look for when we look for solving many of the challenges that we face today. Once again, congratulations, and thank you so much for coming. It was our pleasure to host you here, and hopefully this will not be the last one for us, and we'll look forward to it. Thank you.
This concludes today's competition. I would like to ask the judges and all the finalists to please remain for photos. Audience, feel free to visit the cafeteria just outside this room, or see the visitor's desk for available tours. Thank you, and have a good day.